This is Dr. Vishnu Shankar Sinha, PG Department of Botany, Tata College, Chaivasa. Today I am going to discuss beta oxidation of fatty acids. Fats are important source of energy and constitute approximately 84% of stored energy and 50% protein and less than 1% carbohydrates. Interestingly, 1 gram of fat gives 9 kilocalorie energy. Beta oxidation is the aerobic and catabolic process in which oxidation of fatty acids on the beta carbon atom to generate acetyl coenzyme A releases acetyl coenzyme A, FADH2 and NADH get entered in another metabolic processes called citric acid cycle or Krebs cycle, in which ATP is produced to be used as energy. Beta oxidation occur in the mitochondria in eukaryotes and in the cytosol or prokaryotic cells. Fatty acids are oxidized by most of the tissues in the body, but brain, erythrocytes and adrenal medulla cannot utilize fatty acids for energy requirement. Stages and tissues. The beta oxidation of fatty acids involves three stages. Number one, activation of fatty acids occurring in the cytosol. Number two, transport of activated fatty acids into mitochondria. It is conducted by carnitine shuttle or carnitine carrier system. And number three, beta oxidation proper in the mitochondrial matrix. Number one, fatty acid activation. Fatty acids are activated to acyl coenzyme A by thiokinase or acyl coenzyme A synthetases present in the cytosol. The reaction occurs in two steps and requires ATP, coenzyme A and magnesium ions. Fatty acids reacts with ATP to form acyl adenylate, which then combines with coenzyme A to produce acyl coenzyme A, and ATP is converted to pyrophosphate. This reaction clearly shows first fatty acid is converted into acyl adenylate and then acyl adenylate is converted into acyl coenzyme A. Here one molecule of ATP is involved. Number two, transport of acyl coenzyme A from cytosol into the mitochondrial matrix. The inner membrane of mitochondria is impermeable to fatty acids. With a special carnitine shuttle or carnitine carrier system operates to transport activated fatty acids from cytosol to mitochondria. Cytoplasmic fatty acyl coenzyme A interacts with carnitine to form fatty acyl carnitine in the presence of the enzyme carnitine acyl transferase 1 or CAT1, which is present on the inner side of the outer mitochondrial membrane. The acyl carnitine is transported across the membrane to mitochondrial matrix by a specific carrier protein. Carnitine acyl transferase 2 or CAT2 converts acyl carnitine to acyl coenzyme A, and carnitine released returns to cytosol for reuse. Acyl coenzyme A involves in beta oxidation. This is the picture which clearly shows the uh, entering of the fatty acid from cytosol to mitochondrial matrix by the help of carrier protein and carnitine transport. Beta oxidation proper in the mitochondrial matrix. Each cycle of beta oxidation liberating 2 carbon unit acetyl coenzyme A occurs in a sequence of 4 reactions or steps. So steps of beta oxidation. Step 1, oxidation by FAD by linked dehydrogenases. Step 2, hydration by hydratase. Step 3, oxidation by NAD linked dehydrogenases. And step 4, thiolytic cleavage thiolase. This is the overall reaction of beta oxidation. Now, acetyl coenzyme A is converted into trans-enoyl coenzyme A in the presence of acyl coenzyme A dehydrogenases, and one molecule of FAD is involved. The 2-trans-enol coenzyme A is converted into hydroxyacyl coenzyme A in the presence of enoyl coenzyme A hydratase, and hydroxyacyl coenzyme A is converted into ketoacyl coenzyme A in the presence of hydroxyacyl coenzyme A dehydrogenase, and then generation of the acetyl coenzyme A. The entire beta oxidation can be divided into 4 stages: dehydrogenation, hydration, dehydrogenation by NAD, and cleavage. First, oxidation by FAD. The first step in the oxidation of acyl coenzyme A by the enzyme acyl coenzyme A dehydrogenase. A double bond is formed between the second and third carbon, that is alpha and beta carbons of the acyl coenzyme A chain, entering the beta oxidation cycle. In this step, uses FAD as a hydrogen acceptor and produces FADH2, which will enter in the citric acid cycle and form ATP. This is the reaction where the acetyl coenzyme A is transferred into 2-trans-enoyl coenzyme A and liberate one molecule of FADH2. Hydration. The second step is the hydration of the double bond to form beta hydroxyacyl coenzyme A. This reaction is catalyzed by an enzyme, enoyl coenzyme hydratase, hydroxy group in carbon number 2 in place of the double bond, and this step requires water. Here the trans-enoyl coenzyme A is converted into hydroxyacyl coenzyme A in the presence of enoyl coenzyme A hydratase. Now, third is oxidation by NAD. In the third step, beta hydroxyacyl coenzyme A dehydrogenase catalyzes the second oxidation and generate NADH and produce beta ketoacyl coenzyme A. It is NAD dependent reaction. NADH will enter in the citric acid cycle and produces ATP. This is the reaction. Thiolytic cleavage. Finally, the fourth step in beta oxidation is the liberation of a 2-carbon fragment, acetyl coenzyme A, which enters in the tricarboxylic acid cycle, and acyl coenzyme A. Here, beta ketoacyl coenzyme A is cleaved by a thiol group of another coenzyme A molecule by ketothiolases. The new acyl coenzyme A, containing 2 carbons, shorter than the original acyl coenzyme A chain, that re-enters in the beta oxidation cycle. This process continues till the fatty acid is completely oxidized. This is the reaction. Energy yield and end products. Each beta oxidation cycle yields one molecule of FADH2, one molecule of NADH, and one molecule of acetyl coenzyme A. So the total ATP molecule generated is 17 ATP, and this is the calculation. One FADH2 liberate 2 ATP molecule, one NADH liberate 3 ATP molecule, and one acetyl coenzyme A liberate 12 ATP, hence it is the 17 ATP. In addition, beta oxidation yields great amount of water. This is beneficial for eukaryotic organisms such as camels, given their limited SS to drinkable water. Oxidation of palmitoyl coenzyme A. Palmitoyl coenzyme A undergoes 7 cycle of beta oxidation to yield 8 acetyl coenzyme A, 7 FADH2 and 7 NADH2. This is the reaction where the palmitoyl coenzyme A react with coenzyme A SH and 7 FADH2, 7 NAD and 7 molecule of water liberated 8 acetyl coenzyme A molecule, 7 FADH2 molecule and 7 NADH2 molecule. This is the calculation of ATP. 7 molecule FADH2 liberated 14 ATP. As you know, 1 FADH2 is equal to 2 ATP. Similarly, 7 NADH2 liberated 21 ATP and 1 acetyl coenzyme A liberated 12 ATP. So, 8 acetyl coenzyme A molecule liberated 96 ATP. Hence, the grand total is 131 ATP. There are 2 ATP utilized for the activation of palmitoyl coenzyme A. So the net yield of oxidation of one molecule of palmitate is 129 ATP. Significance of the beta oxidation. The energy liberated during the beta oxidation is used in heart muscles or skeletal muscles. Thank you for watching, and for detail you may consult the Biochemistry of Harper. Thank you.